directly in the pharmaco. So basically, one has to understand that it is not only that the drug regulatory process when you are going for registration and approval, not only the impurities is in focus, but the impurities are focused even in the generic space when the drug even becomes pharmacopoeial. Now, we have another category of guideline which was brought in 2014, which is M7, ICH M7, which is assessment and control of DNA reactive mutagenic impurities in pharmaceutical to limit potential of potential carcinogenic risk. Now, here we have to understand the situation. What is the importance of this guideline? I am taking 2 medicines daily at night. One is for benign prostate. Another is for a little bit of high cholesterol. Although I am able to control largely through lifestyle changes these days. After retirement, of course, when you are in a job, you have to rush for the job in the morning. You get a little time for exercise otherwise, or now I can, after superannuation, I can spend longer time for exercise and walk. Maybe I spend in garden almost 1.5 hours in the morning. So you can control your problems with regular exercising, walking, being active throughout the day. Now the issue comes over here. If I have to take this medicine, I'm taking this medicine for almost now 20-25 years. And of course, prostate medicine is of recent origin that has been added, but cholesterol medicine I'm taking, if I remember rightly, I started in 1995. Now almost 25 years. If there is one brand that I'm using, and that manufacturer is not worried about the mutagenic impurities, someday the doctor may detect that I have been afflicted with cancer. Now, normally you will not realize that why cancer has developed in my body, what had been the reason. Is it the food adulteration, chemicals, spray, pesticides? Or the culprit can even be the lifelong, the drugs you are taking. Because that impurity is always going to your body to a certain extent, because the same company is manufacturing same, and it has its own specification sheet, which is buying the API with that, meeting that specification sheet from the market. So that is the reason that the world has really focused a separate category they have made, mutagenic impurities in pharmaceuticals, to decrease the carcinogenic risk. Now here, very interesting what they have done is acceptable intakes for an individual impurity. Now these guidelines need to be taught to students. I don't know what way, but in the master student, they must be taught. If the duration of treatment is less than 1 month, daily intake is 120 microgram that has been prescribed. If it is greater than 10 years to lifetime, it is the impurity intake should not exceed 1.5 microgram per day. Similarly, acceptable total daily intake from multiple impurities. So if it is greater than 10 years to lifetime, so it is going to be maximum you have to take into the body is 5 microgram per day. Now you may question, I also question to regulatory agencies, why so much stringency and why so much requirement of testing. Other thing has been introduced. When your food, even in caffeine you are taking, it is having a higher concentration of caffeine in your tea cup or coffee cup. And that also is to an extent, certain extent, it is mutagenic if taken beyond a certain concentration. Like same is the furfural. There are a lot of items in the food which are also mutagenic, and but the only thing is drug is to be compulsory taken. The food variety you are taking. Of course, you are taking coffee day and night, day in out, so you should be little worried about the presence of mutagenic components in that. The brand you are taking, you should be selective, which you are going to drink lifetime. But I will advise that you change brands. Now here, what I did an experiment that I took 3 brands of rosuvastatin, which I'm taking daily in the evening. I gave it to my scientists to check for the impurity levels. All of them were meeting the Indian pharmacopoeia. They were all three Indian pharmacopoeia brands, meeting the Indian pharmacopoeia specification of 3% total impurities. But the one product which I was taking already, Rosy Day from US Vitamins, had the lowest level of impurities, and other brands which are cheaper brands had a very high level of impurities. Although total impurities are limited to 3% and meeting the pharmacopoeia specifications. But why not I can be more safer by knowing what are the level of impurities for the drug which I'm going to, product which I'm going to consume, and the brand which I am going to consume for my lifetime. So this is an issue that a common person may not be able to understand, a normal patient who is not linked to pharma. But as a pharma expert, I could get it tested in the lab to understand the impurity levels. So there's a big concern related to impurity level, mutagenic impurity levels in pharmaceutical product. And you all might have heard about nitrosamine impurities. The current issue of the presence of nitrosamine impurities like NDMA, etc. in sartans, ranitidine. And now they are saying that you have to prove absence of nitrosamine impurities in all the API you manufacture. Now, this is all offshoot of the overall change emphasis on focus on impurities. Now, a very important development which has happened in 2016, August 2016 in generic space also, ANDA submissions refused to receive for lack of justification of impurity limits. US FDA has made it mandatory and given a warning to industry throughout the world that if you are defining some impurity limits in your dossier, and if you are not giving proper justification of the limits you have decided to be present in your drug API or the drug products, we are going to refuse, we are refusing to receive even your dossier submission. Now, this is a new thing that has come, refuse to receive. In some other guidelines also, they have put, for other reasons also, they have said refuse to receive if the dossiers are not complying to this requirement, critical requirement. So they say, we will not entertain your dossier. We will reject it if the justification of the impurity limits is not provided in the dossier. So this is become a mandate for industry to comply to these requirements. And we in India and government institutions, private institutions, good ones can take this job, but in government institution, it becomes a more so a responsibility. Why I took this whole field, adopted this field in NIPER. I tell you the story. I joined NIPER as a first employee in 1994. We started building up the institute, creating the labs. I was responsible for all the initial purchases of the equipment. Being a trained person in Punjab University, while handling the projects, I was aware of the government purchase procedures. So I really spent almost 10 crores at that time. So up to 1997, we were building up the place. Once the place and the laboratories were ready, our director, founder director, Dr. CL called a meeting of all the faculty members. He said, friends, this is the date and you have to come out with the proposal, PowerPoint presentations. That time we didn't even use the PowerPoint, it came later. That time it was only the slide projector, transparency sheets. You come out with your proposal of what research you will pursue in NIPER. So I made a presentation on impurity profiling and degradation profiling, and including stability testing. Now, everybody in the house, maybe we were initially core faculty, around 10 core faculty, everybody laughed. What is this science that he's talking about? What is this research area talking about? Dr. Call, to my advantage, had come from industry. So he knew about the relevance emerging because he was in Ciba Geigy, which was an innovative industry company. And he understood the requirement of these impurities, which were existing, but not formally as a guideline, but the requirements were existing that you need to create a API and the product to the highest purity. He said, Fine, Saranjit, I appreciate that you have brought out. I give you all the grants. I give you green signal to continue with this work. You carry out this research, train students, because industry would be needing these students when the industry goes international, global. And eventually today, once he called me, in the evening, I was in my office around 6 o'clock. He called me, Sanjit, 31st March is coming. Government has funds of to the tune of 6.5 crores. Will you like to spend that money? I said, yes, I need equipment, hyphenated tools for my lab. He said, okay, done. Give me a proposal by 9 o'clock in the morning. And by chance, I had given a consultancy to some other company. It was later in 2005-6 that he indicated to me the availability of fund. And I got 5.5 crores, could buy the equipment. Now, because of the requirement of impurity controls, whole of the industry has to do. And it is our responsibility, whether a pharmaceutical chemistry, because pharm analysis comes under the pharmaceutical chemistry. So it is our absolute responsibility that we prepare the manpower for this work for industry. So how manufacturers are supposed to justify limits in existing generic drugs and products? First is, if the compendial products are there, conform to limits defined in pharmacopoeias. Compendium is pharmacopoeia. So whatever limits are prescribed, you can industry can have stringent in-house specifications. But if your products are compendial and you conform to the compendia monograph, that is good enough. Now for the non-compendial products, which are not listed in the pharmacopoeia, because it may be newly out of patent and it has not yet been picked up by pharmacopoeia to develop a monograph. So you have to expected similarity with the originator. So you have to take an originator product and see how much of the level of impurities, store it on stability, and then look for the change that has happened in the degradation products that are produced on storage, and conform whatever the limits are there in the originator products, your product also should meet those. If higher than originator, justification to be provided based on the literature, presence in higher quantity relevant marketed products, presence as a signal in metabolite, or through safety studies. So this is what they are saying, justification for the limits. Otherwise, we will not refuse to receive your dossier. And spectral studies and limits outlined by ICH, FD, and other agencies, the guidelines which I have shown you, those limits that have been defined to 0.1% characterization has to be done, identification has to be done. Now, if I have to do impurity profiling or degradation product profiling, there are a set of studies, analytical activities which have to be carried out. And the set of activities include detection, identification, quantitative determination, and then safety evaluation if it is more than 0.15%. So first is you have to detect the impurities, which are what is the number present, how many are present. Like I showed you, loratadine 3 impurities. So it means the HPLC system which we use, the method we developed allowed me detection of 3 impurities. Now, a very interesting thing which sometimes industry hides. I can go back and show you that chromatogram. See over here. Now, one major defect in this chromatogram is, because we were working in our lab on loratadine, this is a published paper from Dr. Reddy's. They had published this paper and we were working on loratadine. See over here what they have done. One good way they try to represent, because they have only identified these 3 impurities, fine, and they gave the structure of this impurity, elucidated structure through synthesis, their spectral study, chromatography studies, all are included in this paper. But very interesting thing, when we started working in the lab, we found that a lot of impurities are coming initially. I said, where are those impurities in Dr. Reddy's API? Then I critically looked into the whole chromatogram starts from 5 minutes. So what is the status of 0 to 5 minutes? They have hidden all the impurities which were there from 0 to 5 minutes. But you can't escape. In publication that has got escape. But dossier people, they know how Indian people do try to manipulate the system. They will not tolerate your this nonsense type of nonsense 0 to 5. You are not showing any impurities. And I believe me, when we had a project coming from other company on loratadine, there was 7 impurities which are between 0 to 5 minutes. So this is one has to be careful and very precise in developing the methods, chromatographic separation. So you have to detect first is a separation, then through separation you detect them, then you identify, then quantitate, and then qualify. Once only that you have quantitation and you have identified, you will have a pure sample, and pure sample will be subjected to safety evaluation. Now, here, all the ICH guidelines and other things that talk of identification. What is the definitions of identification? So let us look into, first it's what they talk of, ICH guideline Q3AB talk of identified impurity or degradation product. In Q3A, they talk of identified impurity. In Q3B, they talk of identified degradation product. So it is an impurity or degradation product for which a structural characterization has been achieved. But if you look into the dictionary meaning, today we are talking of structure elucidation. So it says identified impurity or degradation product means an impurity or degradation product for which a structure characterization has been achieved. But if I go to dictionary, there is a difference in the definition of identification and characterization. What are those? So let me add a clarification on definitions. Virtual difference between the words identification and characterization. So what is identification? Google says the act of recognizing and naming something. Act of recognizing and naming something. So if I separate on the chromatogram, and if I have standards available, I am able to name those peaks. That is identification. So recognizing and naming of peaks on HPLC chromatogram by using well-characterized standards. This is identification. So what is characterization? A description of the distinctive nature of features of something. Means elucidating the structure. So recognize and naming of peaks on HPLC chromatogram by structural elucidation through spectral study in absence of standards. So if you have standards, you are identifying. If you don't have standards available and there are some additional peaks of impurities, you need to do the structural elucidation. Now, automatically, when I'm talking about impurities and degradation, they are trace components, and you have to characterize those trace components. Give a structural elucidation through spectral. So, what is the importance of characterization and structural elucidation? Let us understand this. Structure knowledge is important for synthesis of the compound. Until I know what is this, if I have not known the structure, I will not be able to synthesize it. Essential for preparation of its reference standard. Only if I am able to synthesize, I will be able to provide a reference standard. Must for quantitative determination limit setting. Until I have a standard, how do I quantitate it? Exact quantitation. Quantitation you can do on peak area comparison basis, but that is not exact quantitation. Knowledge of structure can help in silico determination of toxicity and even physicochemical parameter. We have software available. We can give you toxicity assessment of different variety of toxicities and even physicochemical parameter. We have been using in our lab direct software, TopCat software for toxicity assessment. And then these days in the last period of my retirement, before retirement, 2, 3, 4 years, we start with ADMET predictor. ADMET predictor will give you not only the physicochemical properties, it will give you all ADME parameters. ADMET means ADME plus toxicity. So it can predict all. So then pure compound can then also be used for in vivo safety evaluation. So this is important that we do characterization and give the structure to trace impurities. Now, there are two approaches for the characterization, structure, elucidation of impurities and degradation products in APIs and drug products. Now, why I have taken such an elaborate discussion with you and not going to specific. You must be surprising that Dr. Saranjit is just talking what is known to us. Maybe yes, but I need to emphasize. When we are talking of trace component, the whole technology changes. If you have 10 milligram compound, you can take a tube, NMR tube, and put your sample into that, dissolve in any of the deuterated solvent where the solubility is, take the NMR. So you don't need a modified NMR. You need only a spectroscopy tool. If it is a major component, you do the assay by HPLC or by titrimetric even, or spectrophotometric even. But this cannot be done for trace components that way. Can I use spectroscopy for pure spectrophotometer, UV spectrophotometer analysis of any trace component? No. So the whole approach changes. So what is the difference between conventional approach and the modern approach? The conventional approach is that you have a trace impurity, whether it's an impurity, synthetic impurity, or degradation product, or intermediate, or whatever it is of reaction. And this is a drug. So what you have to do is you go for separation, isolate it, enrich it, and do the synthesis then. So we have to enrich the trace component, reducing down the drug, presence of drug, or eliminate the drug, make it into 99% or 100%, and then follow by a spectral analysis. Now it means I am creating a bulk into milligrams of this trace impurity, converting to milligram of same, and then I subject to spectral analysis. Or I can do synthesis and confirm through spectral analysis by having some idea of what this can be through initial spectral analysis, and then I do synthesis. But up to enrichment, this is enrichment. Now this is a very tedious approach. We have been using preparative HPLC system in our lab for industrial projects. When they send us a sample that please characterize this impurity, we had been using preparative and we were going for isolation till we were not given 5.5 CR grant to us. Now the modern approach is use of all hyphenated tools, LCMS, LCNMR, LCIR. These are all available today. LCMS instruments range from anywhere from 1.5 CR to 7-8 CR. LCNMR again is something which you can buy up to 3 CR. LCIR is again 1 CR. So you do the separation and directly push to MS, NMR, and IR. We have LCMS, NMR instruments which are combined equipment. We have LCIR. Now you'll be surprised. IR is a very different principle. All they could have built the LCIR. There is always an interface in all this. When we call them as hyphenated tools, there is an interface. LC. Suppose you have 1 ml per minute flow rate, and only 0.1 ml per flow rate will go into the mass system. Rest will go into the waste. NMR, LCNMR. There's a total different technique that we have to have a probe coming from down, inverted probe. So we have to have a different method. We can hold the sample into this, or we have to initially prepare the sample, have multiple injection into LC, put them into loops, and then we empty the loop and send to the probe. It's a quartz probe going down below the magnet. And IR, what is done is there's a KBR disc that is prepared, and when LC exudate is coming from LC, the component, the mobile phase, which is organic mobile phase, is sprayed over to on the KBR disc. Because KBR can dissolve in water, so it has to be a pure organic mobile phase that is sprayed on the KBR disc so that the drug or the component of interest is deposited on the KBR. And that is done under some heated environment. That's why it is a closed type of chamber, so that solvent evaporates and your component deposits on the KBR. So 1 or 2 techniques and interfaces have been developed for that purpose also. So we have been using these hyphenated tools in our lab because we got the support from the government. And in 2012, we came out with this review. Those who are interested to know further details about it, a critical review on the use of modern sophisticated hyphenated tools in the characterization of impurities and degradation products. This is a lengthy review starting from 148 page going to 173 page. published in Journal of Pharmaceutical Biomedical Analysis. Why I have written one or two of these reviews? Question is, in India, we were very new to this whole challenge which were coming. Although it has come in late, as early as 1995, 1996, 97, but in India, slowly things percolate into the industry, and challenges when they start exporting, more and more companies start exporting. So my purpose was that let us sensitize not only Indian people, but in industry all over the world, because the conventional way of enrichment isolation was being used. And why not the hyphenated tools? And there was no good review article on this for impurities and degradation product characterization. Now, in this paper, we have given a typical protocol for characterization of impurities and degradation products by modern approach. So you have sample containing unknown impurity or degradation products. We first do literature search. Then LC method is developed for sample enrichment. Impurity may be present at 0.1%. If it is required to be enriched because of its low sensitivity, then we can do enrichment of impurities from mother liquor if necessary, or through stress testing. Then impurity profiling method development, stability in the method development, and redevelopment LC method compatible with MS, NMR, and IR. In NMR, we have to use a phosphate buffer, but in MS, we cannot use phosphate buffer because it is when the heated capillary through which a very small mobile phase is moving from HPLC to mass system. It's a heat going through heated capillary because it has to spray in the vacuum chamber in the mass. So if the phosphate buffer is there, it will tend to crystallize and block that capillary, glass capillary. So that's why we need to have a different set of buffers, volatile buffers in mass, a volatile solvent in IR. When it is LC compatible methods, we need to develop accordingly. Then MS and LCMS studies have to be done, direct injection for drug, because it is available in high concentration impurity DP. We have to go for LC separation followed by HRMS, MS and HD studies. And then we lay down the fragmentation pattern for both and compare. You can do LC-NMR studies. We have to have some enriched sample if required by using solid phase extraction system, column switching, or any other suitable preparatory technique. We can have NMR data acquisition in stop flow loop storage mode. And then we compare the spectra, which is proton, carbon, and depth spectra of the drug and its impurities, and compare them. And then we confirm the structure from even 2D correlation, COSY, HMBC, HSQC, etc. And then LCIR studies, we can record the IR spectra, and then we give the structure to the compound. Of course, this is a costly setup. Have LC-MS, LC-NMR, LC-IR, talking of a few crores of rupees, but very worth it if we have to fast do structure elucidation of impurities and degradation products. So what our experience has been that in most of the cases of degradation products, if the drug, we recently published a paper along with a student, my own master student who's doing PhD in NIPER Hyderabad, where we have identified 16 degradation products on stress testing of drug. So all those 16 were very conveniently identified and characterized. But basically, we were using essentially the LCMS tool. LC-NMR, NMR studies come later. If we have some enantiomeric component and the impurity, then we have to go for NMR compulsorily or LC-NMR compulsorily. So to our finding, LCMS techniques on their own carry good capability to provide unequal structure information on majority of trace to minor pharmaceutical components, although subsequent confirmation through LC-NMR, NMR studies is also expected by regulatory agencies. Regulatory agencies always insist that don't give us only a mass data, but give us also NMR data. You can see the books written over here, characterization of impurities and degradants using mass spectrometry by Dr. Brinder and Pramanik, Mike Ashley, and Chen. So there had been focus on mass spectrometry in characterization. And you see FDA published testing method to provide an option for regulators and industry to detect NDMA impurities. And all NDMA impurities, yes, GCMS are methods can be used, but you see, mostly they are MS-based methods, LC-HRMS and LC-MS-MS methods for quantitation, and this LC-HMS for more of the identification and characterization. So these tools are being extensively used today for any kind of impurity. I'm not talking of inorganics, only organic impurities mitigation products. And we have variety of modern mass techniques used for, now you have to decide and understand what are the tools. Sometimes it's mass spectroscopy vendors, manufacturer vendors, they will supply and give you an instrument, sell you instrument which is getting old, getting out of, you say it is just new range is coming, and the old range they will come and sell you. You are spending one time crores of rupees. So there will be no other chance to buy the mass equipment. So you have to understand the variety of mass equipment available and the nature of work you require. There are instruments which are purely quantitative. There are set of equipment which are used for qualitative purpose and the characterization purpose. So they are different tools. So one has to understand so that you buy tomorrow, if you are government college pharmacy in Karad wants to buy and it has got some money, it has to select the right after discussion with all the faculty members. If you are going for bioanalysis and quantitative impurity analysis, you have to buy LCMS-MS. If you are going for triple quad equipment, if you are going for characterization purpose, you need to buy high resolution mass spectrometer, HRMS system, which we call as a time of flight, QTOF, Orbitap, FTI, ACR, triple TOF, QIT TOF, different type of models offered by different companies. We have multiple stage mass spectrometry, which data will be supplementing to for characterization purpose. We have ion trap, QTAP, LTQ, linear trap. This is quadrupole and QIT TOF. And what we can do is precursor and neutral loss scans by using these equipment, time of flight equipment. We have selected multiple action monitoring, which is a triple quad system, QTAP, LTQ, triple TOF, and QIT TOF can do it. Triple quad is more this approach, SRM, MRM is used for quantitation purpose. And then we can use all for post-on extracting ion chromatograms. We can do hydrogen deuterium exchange mass spectrometry, and then we can have molecular format generator, RDB calculator, and isotopic simulation, simultaneous experiments we can do. All this information, what we get, can provide us to the structure of the compound. And we had been using QTOF, LTQ in our labs for the purpose of characterization. And this because a lot of work we have done on mass spectrometry, characterization of impurities and integration products. That's why came out the review in 2014. Critical practical aspects in the application of liquid chromatography mass spectrometric studies for the characterization of impurities and degradation products. Typically, how practically you should approach towards solving the problem through LCMS. Now, these are readable. You should rather, if possible, PCI syllabus allows you to do that. Every student must go through these papers as a part of the training, teaching, or after. Even master students, you people can take this paper and discuss in the evenings with the students and let them give them assignment. And one student brings, studies the paper, makes a presentation and comes before the house. We had been doing this in the evening because all our students and IPER have been the resident students, hostel resident students. They used to sit at 8 o'clock after the dinner, and then 10 o'clock we used to discuss all these threadbare. The line by line we used to discuss and make the training, or batch after batch students. Now, what is the benefit of accurate mass, HRMS? Now, if in a structure we have CH2, it gives me 14.0157 accurate mass. Nitrogen accurate mass is 14.0031. If I only buy an equipment which gives me a nominal mass, I cannot differentiate whether in the structure it is CH2 or nitrogen. But if I bought a HRMS system, which is a high resolution time of flight system, I can differentiate and tell you whether this structure has a nitrogen or the CH2. When I add the accurate masses, so based on nitrogen, again, there is a nitrogen rule also. So based on those all concepts, I can really assign the structure whether it has a nitrogen or CH2. Now, very interesting situation we got in our lab when we did the HRMS studies up to 4 decimal points. You can see over here, this is a mass chromatogram which shows 425.1534 and 425.2536. If you have a nominal mass equipment, it will show you 425. This also will show you 425. Incidentally, this compound has a fragment of it coming, which is giving 407 as a major one fragment. And another, this drug also has a 407 as a, but you can see over here 1425. This is 2424. So here it is, this formula. And if I take this compound and put to the elemental composition calculator, it gives me a different formula, entirely different formula. Only this 1534 and 2538, 4 decimal point difference gives you entirely different formula. And if you look into, this is aprosartan drug and this is a statin drug. Now, this is difference that you can easily come to by having a high resolution mass spectrometer. This is a very interesting example. Here we don't have any nitrogen, but here we have nitrogen, 2 nitrogens in the structure. And if I take 23, carbon 23 at 24, N244S and add up, it gives me 425.1530, which is very close to my experimental value of 425.1534. Here, one thing you have to make sure yourself that the instrument is thoroughly calibrated. Otherwise, you will not be able to get this accuracy. Here also 0.2534 and actual value we got was 425.2536. So you are sure of a structure. An elemental composition calculator will give you what exactly is the molecular formula. In all the synthetic laboratories where we used to use the elemental composition calculator, no, we used to use CHNS analysis. We used to have elemental analyzer. Those days are gone. Nobody is using the elemental analyzer anymore. It is always HRMS data, which is required even to publish a paper for synthesis, and you have done a synthesis of a compound and you have to give the exact value of that. What MSN studies do? MSN study help us to draw the fragmentation tree. What we do is we pick up A, we pick up X, then further fragment it. So what we do is we push this fragment of the main component, that is a molecular ion, put into, whether we do in the positive ionization mode or negative ionization mode. We take this component. This is one fragment, this is second fragment. We take this fragment, put into the ion trap. MSN studies are done through ion trap equipment, put into the ion trap, then bring it back to the quadrupole. And then we are able to see the fragmentation tree one by one, MS2, MS3, MS4, MS5, up to MS9 we can give. So I come to know which is fragment, which mass fragment is coming originating from which parent, which is a precursor for B, which is precursor for D, which is precursor for J. So J is coming through both the roots. I is coming through this route only. So similar is this way that I can get for other major fragment coming from the molecular ion. And then HT exchange studies will indicate to me whether there is nitrogen, oxygen, or sulfur where we have hydrogen, which is a replaceable hydrogen by D2O. So we have hydroxyl, amines, thiols, acids. So they all have a replaceable hydrogen. So exchangeable hydrogen. That can give me more confidence about the structure I've elucidated. Now we published this paper in Analytical Chemistry, very high impact journal, practical and economical implementation of online HG exchange LCMS. Ravi Shah is now in NIPER Ahmedabad. He is associate professor in pharma analysis there. So he was doing PhD under me, and these are all students of mine at master's level. And these are the people who later Ravi Shah joined the Bristol Myers, Biocon Bristol Myers Squibb R&D Center at Bangalore. So these are the two leaders from there. So we had a collaborative publication. So this is a strategy for characterization development in our lab, that we do drug direct injection, MSN studies, MS top analysis, HTG exchange. We collect this whole information. And trace component, if there is, we do online LC separation instead of direct injection, then do MSN study, MS-TOF analysis, online HCD exchange. So we dwell fragmentation pattern of drug and trace assignments of structure to trace based on comparison mass spectra and fragmentation pattern of the drug, and justification through mechanistic explanation wherever possible. So we have handled more than 50 projects on impurity characterization. We have handled almost total 100, but they were not all of them related to characterization of impurities and degradation product, but none of them were published because they were CDA confidentiality agreement. All studies were done under confidentiality agreements. But we have extensively published on degradation products because of our projects that were assigned to the students. So there are a lot of publications you will find that we have been using all these techniques, LCMS-TOF, MSN, and online HD exchange, LCNMR, to characterize irbesartan degradation products. Again, for telmisartan, this is valsartan series. One PhD thesis of Ravi Shah himself was on sartan series and statin series. This is a statin series. So each students were characterizing, handling almost 4 or 5 drugs and doing PhD and characterizing their degradation products. Further, Sunny Piyush Bhardwaj is in US these days. He did on enalapril, fosinopril, benazepril. Mallikarjun did his PhD, he did his master's, and then he did on all pril series. One student, Mulchand Kurmi, did publish 11 papers we published on stability behavior, antiretroviral drugs and the combinations. So one thesis, 11 publications in international journals. This is the 6th in series. Then we have 5th in series, and this is the 10th in series, all which were into antiretroviral drugs. This is other series of papers that we publish, and there are more 25 of them, which I'm not listed over here. Now, we also extended our work to characterization of drug-drug and drug-recipient interaction products. And you can see this paper. This is LCMS investigation of the polypills, which are 5-6 drugs. This was Dr. Reddy's projects. And separation active component classification of the interaction degradation products. Vijay Kumar did his PhD on polypill under Dr. Reddy's sponsor project. Polypill for the renal cardiovascular part 2 LCMS-TOF characterization interaction degradation product, lisinopril and aspirin and mechanism of formation thereof. And this another paper comparative retinol with excipient LCMS characterization degradation interaction and mechanism of their formation. You'll find that I'm talking of 2 drugs only. Atenolol with excipients, and in the previous one, atenolol and lisinopril and aspirin. When it went to further 5-6 drugs, we were forbidden by the company to publish papers further, because that was leading to polypill with the 5-6 drugs. Now, Tarun Handa did on combination of some of the cardiovascular drugs, characterization of a new degradation from nifedipine formed on catalysis by atenolol. A typical case of alteration of degradation pathway of one drug by another. Total route is changed when this is a drug-drug interaction problem. And this is also characterization of interaction for lamivudine and tenofovir in the part 2. Part 3 is interaction products of amitriptyline and tenofovir. And this is interaction products of zidovudine and efavirenz in 11th paper. We have 12th getting ready. So 9th was stability behavior antiretroviral drugs and combination identification incompatible excipients. Multiple drugs we took and did that on 9th paper. So there's no end where you can use the techniques and publish papers. Every paper is accepted very well. I will give you very quickly what is a whole one paper on a series. The number 10, I am going to talk in detail what is the kind of scientific input required when we do all these characterization studies on degradation products. And then we initially go determine the admit properties. Because now every regulatory wants to know what kind of degradation products were produced and give us the tentative toxicity data, whether it has a higher toxicity than the drug, whether it has a carcinogenicity, mutagenicity or not, degradation product, specific degradation product. So we have to give now all the admit properties and the physical chemical properties and the even all pharmacokinetic properties. So this was on fosamapiravir. that we did the study. Now, first we have to go for HPLC. We do the studies in different acid and base, and I'm not giving the oxidative and others, but I'm showing you that 6 degradation products got separated under acid and base conditions. So this is where we identify, not identify, just label these degradation products. Then we do generate MSN data from where the fragments are, which fragments are produced from MS2 fragment of 586. These are the fragments produced. Now we will pick up 498, what fragments are produced. Then we have taken 488 and then 431 we picked up, 429 we picked up. Then in MS4 we took 418 because 418 was produced from 488. So it went down to MS4, MS3, we did all this. So this way we go up to MS9, you can see over here, to build up the fragmentation tree for drug first. We take the accurate mass also, all the drug accurate fragments and their accurate masses we take. This is the exact mass of the probable structure wherever we took. It didn't appear in, it came in nominal mass, but didn't come in, it was seen in MSN study, which gives us nominal mass, but it didn't, some of the fragments didn't appear in HRMS based on the instrumental sensitivity differences. So this type of data is to be generated, which is the exact mass, error between the experimental mass, accurate mass, and the best possible molecular formula for each fragment. These are not degradation, these are mass fragments. Then error, then RDB value, ring and double bonds, possible parent fragment from where it is originating, mass difference from the parent ion, and possible molecular formula for the loss. So this way, we have to sit down and write down the fragmentation tree, total fragmentation tree of the drug. Now, Dilip, due to his talent, he initially had worked in BBRC, then went to Sandoz after doing PhD. And last week he has reached US, working in Washington State University, has been invited as a postdoc and given he made the lab manager of the mass laboratory over there. So people are taking students by name because this is a training that industry needs now, or the institutions abroad need now, and they are taking away the students. And we do collect NMR data, proton, carbon depth data on drug first. Then we do HRMS data for degradation products 1 to 6, all of them, their data. And then NMR data on the degradation products, DP1, DP2, DP4, DP5, DP6. Now DP3 is not included in this list because here we had to go for LC-NMR. Here we could isolate them for NMR studies. So what we do is we re-inject, continue multiple injections we give in the analytical HPLC and collect the exudate and evaporate it. So if it is possible to collect, we go for NMR, otherwise we go for LC-NMR. LC-NMR has a difficulty, DP3, because DP3 could not be isolated. And in NMR, we have residual solvent signal suppression, ACN suppression, and this is the residual solvent signals. So here it is only possible if we can get good data beyond 1 to 4 ppm, or let us say 1 to 3 ppm, we have a lot of solvents coming. Beyond 4, it is much easier to assign the values. So this is a limitation of LC-NMR because of the solvent that need to be suppressed. And this is what we have given the degradation pathway and mechanism of hydrolysis of the drug. So you have to propose in your papers complete mechanism of the formation of different degradation products. This is what is appreciated by regulatory agencies, this kind of complete scientific study towards the characterization. And a lot of industry people have complained to me rather, sir, US FDA auditors, when they come to our companies, they carry your papers and tell us that please look into whether your methods are able to separate out these reported degradation products in literature. So the advantage also is there, but disadvantage has come that if something has been reported in the literature, industry has to revalidate its method for those degradation products which are reported in the published papers. But the benefit would have been, industry benefit is that In ICH guidelines and stability testing guideline Q1A and also WHO guideline, they are talking of stress testing. Stress testing means you force decompose the drug and try to identify the degradation products. It says for an API, the following approaches may be used. When available, it is acceptable to provide the relevant data published in the scientific literature to support the identified degradation products and pathways. When no data are available, stress testing should be performed. It means large part of generic industry, if I publish one paper, let us say on any drug, and India, there are 20 manufacturers, they don't need to do this basic study. They can just provide relevant data published in the scientific literature. Of course, the regulator is asking that if these 5 degradation products are produced, reported in the literature, now your method should validate for these 5 degradation products so that there is no surprise when you put the product on stability. So this is what we say, quality by design. You build quality and don't test the quality, which Professor Boop will be talking to you in detail. Now, I am taking a few minutes more to discuss with you a case study of a project from a pharma company. I can name the company. It was FTC Limited, which is into eye drops. Majority of eye drops come from FTC. So they had an eye drop, which was comprising of ciprofloxacin and dexamethasone. 2 drugs in the eye drop, and they had put preservative benzalkonium chloride. Now, very interesting. They had a one new peak. I'm sorry, I think somewhere the shifting has happened. This is a peak for, this was unknown peak. They said, sir, it may be impurity, it may be degradation products, but we have taken the pure ciprofloxacin dexamethasone we used in preparation of this ophthalmic formulation. But there is a new small peak that is emerging on stability. And most likely it's a degradation product. Can you really help us identify this small peak? Now, this is benzalkonium chloride, this is ciprofloxacin, this is dexamethasone. Now, what is the best way for us to solve this problem? I can do this study, whole study will require, first what I have to do is I have to take on my HPLC system and see whether this peak is rising, this small peak is rising on my HPLC system, because this is the chromatograph supplied by the company. So I had to see whether this peak, small peak is rising and showing its head on my HPLC system by the method which is given by them. Once it comes over here, I develop a method which is mass compatible. I take it to mass to find out whether this degradation product is related to dexamethasone or ciprofloxacin. So which I have to have pure ciprofloxacin in hand and dexamethasone in hand. So I took these 2 drugs from the company. They sent me the samples. So I took the mass study we did on dexamethasone pure, we did on ciprofloxacin pure, and then we did on this small LCMS, we did the study on mass studies on this degradation product, supposedly a degradation product. Now, when I do this, degradation product is showing me peak of 375, 355, 330 fragments, mass fragments, or 319, 309. They exactly match with dexamethasone. You can see over here, 355, 355. It is 337, 337, 319, 319, 309, 309. So just having the mass 3 injection is the mass spectrometer, 2 direct injections of 2 drugs and 1 through LCMS. When we put to mass, we come to know that it's a dexamethasone degradation product. So here, what is the change that has happened? This is 393 and this is 375. It means 18 loss has happened. There is a loss of water. Okay, 375, 393 minus 18 is 375. 18 added to 393. It means there is some loss in degradation product of water. Now we look into the structure of dexamethasone and what is the possibility of loss of water. We have OH over here, CH2OH over here, where the loss is happening, or some other change is happening. So what we do is we take the mass fragmentation, we build up the mass fragmentation tree for dexamethasone first, and then we'll come to degradation product. So what we do is we build up this thing. Blue is, this is whole for dexamethasone based on MS2, MS3, MS4, MS5, MS6 or MS5. We take dexamethasone and build up its mass fragmentation pattern. And we see over here that there's an elimination of water from here in MS2, and there is loss of HF from the drug over here to generate 373, and 375 is due to the loss of water. Now we tried dexamethasone. We take the same degradation product and try to fit in 375. Here also we get 375 if we have this loss of water. It means there is a loss of water happening. So here you can see that this 375 is again with the loss of water over here. We try to fit into all the fragments and we are able to give the same structure. And when we overlay the two, drug to degradation product on loss of water, we can say that there is a parallelism in the drug and degradation product, that we could build up the mass fragmentation 355, 355, 373, 375, 337, 337. Just with the degradation product have this structure minus CHO. So loss of water, where it is happening, it is restructuring itself. This whole moiety over here, change is happening to degradation product in this manner. Now, why this manner will come to you later. Why it is not simple elimination of water from here. Why this change has happened, aldehydic structure, why it has got generated. So this fragmentation pattern was supported by NMR and IR. Then we took isolated this product, and then we could come down and explain to the industry. So what we do is we took the degradation product and generated in the pure form. 10 milligram solution dexamethasone acetonitrile 0.1 normal HCl was kept at 80 degrees. The changes of monitor HV and the reaction was stopped and sufficient conversion was obtained, and we got it into the pure form. So we got a pure form, and we spiking study when we did the same isolated degradation product, which we have isolated. We spiked over on the company's chromatogram behavior, and it was giving me the rise in that. It means whatever the degradation product we did to generate, it was same as it was same, falling on the same impurity. Now we did the IR spectrum of the isolated degradation product, and there was a carbonyl in the dexamethasone, which was C20 carbonyl leading to aldehyde in the isolated product. This product we could isolate easily, and this we took IR, and then there was an aldehyde peak in proton NMR. So 13C NMR, we have CO signal, and there was a new signal in the degradation product. So there was in me some change has happened over there. So based on this, this is dexamethasone. Now, when it leads, water is eliminated, and my student came running to me, sir, there are two tautomeric structures possible for this degradation product. So how to assign the exact structure to the impurity? So we went over to the mass, looked into its mass fragmentation pattern and possibility of which structure will give us the same mass fragmentation pattern. More so, what was interesting was that this impurity was generated on the right side of the two drugs. This is ciprofloxacin, benzalkonium chloride, ciprofloxacin, dexamethasone, and this is the impurity. So it means this impurity was non-polar than the drug, dexamethasone. Had it come on the left side, it would have been more polar. So I asked him to calculate C log P values for both the tautomers. So you can see over here, this C log P value for this compound with this tautomer is 0.988, and this tautomer is 2.858. And this C log P is 1.7852. It means this is the tautomer practically in that small peak that was present in the sample sent to us by the company. So this is the structure which we could give over here. And based on that, we built up the fragmentation pattern, which was almost parallel to the drug, and we could fit into this. So the company, based on the scientific data we provided, we charged 2.5 lakhs for this much work. It was all intellectual work based on the data we received from the mass spectrometry data. We charged 2.5 lakhs, finished this project within 2 days. So, and we earned that 2.5 lakhs. So you spend crores of rupees, you can earn crores of rupees, but having the right scientific temperament, you can do that. So we sent pure compound, and the company people submitted this data to, because this query had come on identification of the unknown peak from US FDA, where this company FTC Limited sought to market this combination of ciprofloxacin and dexamethasone ophthalmic product. So when this data was supplied to US FDA, they were very happy to accept this data, and they issued the approval to this combination of FTC. So as a national institute, NIPER, we had some responsibility of helping in industry to go abroad. And this is what we were able to serve our industry. So I summarize our successful journey from a government institute. That is, working on trace component characterization using hyphenated tools proved hugely beneficial for the institution, myself and my students. They got training, they got good jobs. I got good recognition through our publications. I got editorial assignments due to that. And there have been flow of publication and industry sponsored projects from India and abroad. Some of the projects we did for foreign industry also. Leading industry in India has been keeping track of every student passing from the department who always have been hired by name. I want Dilip, I want Bhupinder, I want this person. We were first to extensively use such sophisticated tools for the purpose, so laid down protocols which are now being followed globally. I see publication after publication now coming, and we were the pioneers in this to initiate this activity in NIPER and issue lot of publications, make lot of publication based on that people in industry all over the world could come to know that how sophisticated tools can be used based on our reviews and the actual publications. But each publication you look into is highly extensive data and lot of intellectual input. I never use software for giving structure to any fragment. Always student has to think over. And there had been a situation that one fragment structure was not justified. And we found that the degradation product structure was wrong that we were visualizing. Only one single fractal out of 40 fragments, mass fragments, one structure was not 100% fitting into the scheme of things. So had been helpful overall to global and industry in meeting stringent regulatory challenge of analyzing trace components. Now, I will say that it's a big opportunity waits every one of you as the requirements of trace component analysis is becoming, requirements are becoming more stringent and expanding globally by each day. So my best wishes to you all. I am retired now. Somebody of you people have to take the lead. Ravi Shah at NIPER Ahmedabad now has world-class equipment, latest models. So he has got into it. He is full of industry projects. He says, I am so much loaded. I have so much teaching load. I have so much administrative responsibility. I'm over that. I am neck full of company projects, industry projects with respect to isolation, characterization, or characterization of impurities and degradation products. So thank you, friends, and Jai Hind. The time, my watch says 11:57. A few minutes for question before handing over to Professor Bhup. I think he must have joined by now.
Yes, he's there.
Thank you so much.
Yes,
thank you for patience listening.
Thank you, sir.
Okay.
Osmani, sir.
Stop sharing now. If any questions are there,
Yes,
then
sir.
I can...
Yes, sir. Yes, sir. Please stop your sharing.
Okay, dear.
The participants, if you are having any doubts, please ask.
Please come over. You are the future of country. You are the future of students also. You train well, teach well. The students gain benefit.
Okay, sir. Shall I proceed?
Please.
Yes, sir.
Okay, thank you, sir. Once again, a good afternoon to everyone. So we have reached the end of this informative, interesting, and an out-of-the-box session on application of hyphenated chromatography and spectroscopy tools in structural elucidation of trace components in pharmaceuticals. In a nutshell manner, sir has informed us the types of impurities, degraded product, and the qualification threshold as per ICH guidelines for industry. Also, we were enlightened the controls given in a various pharmacopoeia with many important tips regarding the reporting different individuals and multiple impurities in different brands, parameters the manufacturer need to mention regarding the impurities, and the manner of dossier writing in some companies. In a brief summation, Dr. Saranjit Singh sir has explained the concept of the gray and novel area in hyphenated chromatography and spectroscopy tools and the hardship faced during application of the same. Also, the experience of initial work at NIPER mentioned by sir will surely reap the innovation in us to get additional exposure or recognition for our institute at national as well as international platforms. With this, I am thankful to Dr. Saranjit sir for taking out time for his busy schedule and accepting the invitation as a speaker for this webinar. Also, I would like to extend my thanks to our respected principal sir, Dr. Cherde sir, who is the coordinator of this program, and the organizing team who are working from the last few days to make this webinar a successful event. Thank you very much, sir.
Very nice connecting with you, Professor Saranjit. You know, it was.
How are you,
Sir,
Parvati?
please accept this virtual certificate
Thank you, Ji.
from us.
Good. Excellent.
Sir, thank you very
It is the sun.
They all were very fortunate to have heard from our legendary, world-renowned speaker, Professor Saranjit Singh, and who has ventured into this novel area, you know. Very successful man and very dedicated personality and very respected personality across the world. And you all are privileged to have heard it from you.
It's our pleasure that all the time he is with us. Thank you very much, Saranjit Singh sir. Thank you. Thank you.
Bye-bye.
Shende sir, I
Should-
think.
Should I share
yes.
screen?
Sir, 1 minute. 1 minute.
Okay, okay.
Sir, next speaker is with us. So I request Shende sir to start with the session.
Okay, sir. Thank you, sir. A very good afternoon to all participants, my colleagues. I am extremely privileged to have with us eminent personality in pharma field, acclaimed globally for his scientific research work, Dr. Bhupender Singh Bhup. Before proceed, as a formal part of this program, I request Dr. M.S. Cherde sir to welcome Dr. B.S. Bhup by virtual for bouquet.
Sir, welcome, sir.
Wow.
Thank you, sir. It's a great pleasure for me to introduce our guest speaker, who is going to talk about accomplishing pharma excellence and regulatory compliance using modern and systematic QBD paradigm. Professor Bhup has contributed 4 decades of dedicated service towards pharmaceutical education, research, and industry. He is working in the domain of pharma QBD and related paradigm, biobehavior, and nanomedicine. Dr. Bhupender Singh is currently working as a professor in pharmaceutics and biopharmaceutics, affiliated to University Institute of Pharmaceutical Sciences, Punjab University. He has guided over 110 researchers, including 35 PhDs and 9 postdoctorates. He has completed 6 industrial consultancy assignments and has 8 patents to his credit. He have 3 technology transfer of nanotechnology based drug delivery products and 15 computer software to his credit. He has earned research grant over 7.30 crores from governmental and corporate sectors. He has to his credit over 400 original paper publications, including 16 books by prestigious international publishers and 81 books chapters with H index of 53 and citation of over 11,000. He has delivered more than 350 plenary keynotes and invited talks in India and overseas, including US, Canada, UK, and many more. He has pharmaceutical consultant and eminent trainer for industrial scientists from leading pharma companies in India and abroad on different fronts. Dr. Bhupender Singh has received several awards and accolades of for the contribution to scientific community, in which major light leading Educators of World in 2007, Pharma Buzz Pharma Personality Award 2008, Shiksha Ratan Puraskar in 2008, Innovative Scientist Award in 2012, Pharma QBD Outstanding Performance Award in 2014, Bharati Vidyapith APTI Pharmacy Teacher Award, Scientist for Excellence Award, Innovative Healthcare Researcher Award, Global QBD Excellence Award, Elsewhere Best Research Paper Award in 2020 and 21, Pharma Excellence Award, so like many more. His expert panel on UGC, NBA, NAC, AICT, PCI, DST SERP, DBT, UPSC, PPSC, IPA, APTI, SPDS, and Commonwealth Secretariats. To promote pharma profession, he served as a chairman, University Institute of Pharmaceutical Sciences, Punjab University, fellow and member, University Senate and Syndicate, Dean as a Dean of Faculty of Pharmaceutical Sciences and Alumni Relation. He is a founder coordinator of National UGC Center for Excellence in Nano Biomedical Applications. He has recently added on among top 2% scientists across the world in 2020 as well as 21, University USA. So this is very brief introduction, sir. Professor Bhupendra Singh has very much research interest in vast area and have active association with different societies, score of prestigious universities and institutes in India and beyond. So with this, please welcome and please join me in welcoming our guest speaker. So please, Dr. Bhupendra Singh, to take the charge of this session and enlighten us. Thank you, sir.
Welcome, sir. Sir, you are muted.
Am I audible?
Yes, sir. Yes, now you are audible.
I think I must share the screen first.
Yes, it is visible, sir. Your screen is visible.
Okay, screen is visible. Thank you so much. Thank you so much, Dr. Manoj Erde and Dr. Shinde for having me provided with this opportunity to speak on a different topic, although the conference faculty development program of this government teachers, government pharmacy college teachers is on a, a very broad and but different paradigm. But Dr. Manoj was so persuasive and so respectful in inviting that you should speak on this topic, which is relevant in the modern era and which the participants, the faculty participants are eager to know and understand. So, my dear friends, I'm, as Dr. Shinde has already, informed you in this nice introduction, that I'm going to talk about accomplishing pharma excellence and federal compliance using